Hello students, welcome back to my channel Competitive Botany. In this video you are going to see the concept of linkage, coupling repulsion and also cis and trans configurations. Okay, so in the previous class we are already studied about discussed about the coupling and repulsion phases of linkage. So what is linkage? First you should know the linkage is nothing but the total genes present on single chromosome they inherit as it is to the next generation. They do not tend to separate during the gamete formation. This phenomenon is nothing but linkage. So the chromosomes, so the genes present on single chromosome, they do not tend to separate during the formation of gametes. Okay, so as it is, the parental combinations will go on to the gamete formation, will simply go to the gametes. Okay, so then the parental combinations are produced more in the next generation. That is called linkage. Okay, that is called linkage. So let's see the Morgan's concept of linkage. T.H. Morgan, he is working on Drosophila. At that time he did not know completely about coupling and repulsion phases, but he suggested that two genes are found in coupling phase or repulsion phase when they present on single chromosome, same chromosome. That means if two dominant genes present on single chromosome, the two dominant or two recessive genes are present in a single chromosome, so they are showing coupling. That means they went to gametes as it is without change. So that is called coupling phase. We are already studied in the previous video. Okay, so if one dominant and one recessive gene is present on the single chromosome, then they repulse each other. That means they tend to separate each other. That phenomenon is nothing but repulsion. That is also studied in the previous video. I think you understood very well. Okay, so according to Morgan, the degree or strength of linkage depends upon the distance between the linked genes in chromosome. Okay, that means, so the genes present on single chromosome, they do not tend to separate. They went as it is. Okay, so they reach as it is to the gamete without change. This is called linkage. Okay, this is the phenomenon called as linkage. So here Morgan says that the strength of linkage depends upon the distance between the linked genes in a chromosome. That means, so the distance between the, if imagine these two are the A, B, C, D genes, these four are the, okay, so the distance between two genes, okay, so the linkage depends upon the distance between two genes. That means here the distance between A and B genes is very less. That's why here they are having more linkage capacity. They are showing more linkage. Okay, so that is only he is telling. Okay, so but the linkage phenomenon in between A and D is less when compared to the A and B, because the distance is far from A and D when compared to A and B. Okay, D gene is far to the A. That's why the A and B showing more amount of much linkage. Okay, when compared to A and D. Remember. Okay. Let's see the chromosome theory of linkage. So this was proposed by the scientists Morgan and Cassill. This theory is also known as theory of linkage. So it states that genes present in the same chromosome are inherited together is called linkage. Okay, so they are inherited together. That phenomenon is called as linkage. So you know that the linked genes arranged in linear manner. as I shown you before, they are arranged in linear manner on the chromosome. So the degree of linkage is determined by the distance between two genes. That means linkage strength is inversely proportional to the distance between two genes. That means if the distance is low, the linkage is more. If the distance is far, then the linkage is less. Okay. So as I already told you before. Okay. So linked genes show two types of arrangements in heterozygous individuals. Let's see the first arrangement, that is cis arrangement. Second one is trans arrangement. Let's see the first. So this arrangement is called as cis arrangement, or this is called cis heterozygote. Why? Because two dominant genes are located in one homologous chromosome. These two are the homologous chromosomes. So this is the location of that particular gene. This is also the location of that particular gene. Okay. Two dominant genes were located in the same chromosome. That is called cis configuration or cis arrangement or cis heterozygote. So in the same way, another homologous chromosome having small a gene and small b gene. Okay. So this is called heterozygote. Okay. That means two different genes are present. That's why this is the hybrid. That's why heterozygous condition observed. Okay. So one chromosome having dominant alleles, two dominant alleles, one chromosome having two recessive alleles. These two are the homologous chromosomes. They are in pairs. Okay. So let's see the next configuration. Before that, this arrangement is called as cis arrangement. The heterozygote with such arrangement is known as cis heterozygote. Okay. In this case, one homologous chromosome containing one dominant allele or one recessive allele. Another homologous chromosome containing one recessive allele, one dominant allele. So this is also heterozygote. Why? Because two different genes are present. Okay. So but the arrangement is different. Here one dominant, one recessive. Again one recessive, one dominant genes are present on individual chromosomes. This type of arrangement is called as a trans arrangement or trans configuration. This heterozygote is called as trans heterozygote. So they were in repulsion condition. Cis configuration chromosomes, they are showing a coupling phenomenon, but trans arrangement heterozygotes showing repulsion. Okay, repulsion. That means they tend to separate. These are not tend to separate, but they went as it is to the next generation, but they are showing separation during the formation of gamete. Okay, these two are separated, these two are separated. Why? Because one is dominant, one is recessive, one is recessive, one is dominant. That's why. Okay, so this is called cis arrangement and this is called trans arrangement. Okay. After that, we should know what are linkage groups, where they are located. Okay, so every individual having linked genes. Okay, every individual, the whatever chromosomes present on a single, whatever genes present on a single chromosome, so they are showing linkage. This is a common phenomenon. Okay, so that's why we are getting more parental characters than the recombinant characters. Okay, the total genes which are located on the chromosome constitute a linkage group. You know that the total content of genes, they are showing linkage. They do not tend to separate, they went as it is to the next generation. That is called linkage phenomenon. The number of linkage groups of a species is equal to haploid chromosome number of the species. Why? Because homologous chromosomes showing identical genes. Okay, homologous chromosomes showing identical genes. Okay, that's why. Okay. So let's see the examples for linkage. So if you see the Drosophila, the chromosome number is 8. The haploid number is 4. 4 pairs of chromosomes are there. That means 4 pairs of homologous chromosomes are there. That's why 4 linkage groups observed in Drosophila. If you see Homo sapien man, you will observe 23 pairs. That means 23 linkage groups observed in human. If you can see Siamese, Siamese containing 10 pairs, that means 20 chromosomes. That means 10 linkage groups observed in Siamese. Okay, in the next video I will upload regarding the types of linkage. Okay, so please watch that video too. Thanks for watching.